Summer, grab the people in the waiting room too. We always have latecomers for some reason. Welcome to another ALK Talk, and we really have a special guest here. And I'm not going to be doing too much talking because my throat's killing me, but I am going to launch some polls here real quick. So the first poll question is, what type of treatment are you on? This all will give our guest, Dr. Schneider, a chance to see who's in the audience. And I'll launch this pretty soon. I'll share the results. Okay, I'm in the poll. Share the results with everyone. Alectinib is 63%, and then 13% is lorlatinib, 7% is brigatinib, and I believe we have a few people on chemotherapy. So go back to the next question. Is your length of diagnosis? Okay, and the poll. Share the results. So we got 2 and under. I'm going to lump you guys together. And that's rough about 53%. And then 3 to 4 years is 26%. And 5 to 6 years is 11%. And I'm stop sharing that. And I'm going to turn it over to my co-host, Summer Farman.
Hello, everyone. Welcome to ALK Talk. Thank you for joining us. So happy to have you here. Thank you for supporting our kickoff of ALK 101 last week. We were so excited. We had a great turnout. And if you have not seen that yet, you can catch that on YouTube. We have some very special guests here tonight, and moderating our talk will be Emily Venanzi and Marc Muskavitch from our medical committees. And then we have the Lung Cancer Research Foundation representatives and Dr. Schneider herself. So Emily, why don't you kick us off?
Hi, everyone. It's really a privilege to have Dr. Schneider here tonight to talk about her research. And we have awarded her a research grant through a new partnership that ALK Positive has with the Lung Cancer Research Foundation. And I'm going to talk a little bit about that. And Colleen from LCRF will also talk to you about our partnership as well. It's been a privilege for ALK Positive to partner with the Lung Cancer Research Foundation in co-funding 2 ALK research grants this past year. And these have been made possible in large part by the fundraising and donations of so many of you in the ALK Positive community. So thank you so much for your efforts and for your generosity. LCRF's mission is to improve lung cancer outcomes by funding research for the prevention, diagnosis, treatment, and cure of lung cancer. And they've been focused on funding cutting-edge lung cancer research for about 20 years now, using a rigorous scientific review process to select their grant awardees. And we at ALK Positive have been fortunate to benefit from their expertise in selecting these ALK research grants. And I'm going to briefly walk you through the process that our 2 organizations use to work together to select which research to fund. So I have one slide here. Okay. So to start the process off, a group of us, including members of the ALK Positive Medical Committee and the LCRF Scientific Executive Committee, first worked together to write a request for applications, which went out to the scientific community last year. And this RFA detailed the kind of projects we were interested in funding. And that also said that we were looking for projects with the greatest potential impact for patients. And in response to that request, we received 13 letters of intent, which are like shorter pre-applications describing what each applicant wants to research. And an expert panel reviewed the LOIs. And on this panel were both members of the ALK Positive Research Review Panel group and members of the LCRF Scientific Executive Committee, who are knowledgeable about ALK lung cancer and ALK research. And the panel met and together chose the 6 best LOIs and asked those applicants for full proposals detailing their projects. Those full applications were then reviewed by the same expert panel, and the panel agreed on 2 projects they thought were both the strongest scientifically and also the most likely to have an impact for patients. And then finally, the boards of directors at both ALK Positive and LCRF learned about and approved the awards. And both boards were really enthusiastic about both projects. And we're excited to hear about how they progress in the next 2 years. And one of those awardees is here with us tonight, Dr. Jamie Schneider, and the other awardee is Dr. Alvaro Quintanal Villalonga, and he is from Memorial Sloan Kettering Cancer Center, and hopefully we'll hear from him in the future. So now I'd like to hand it over to Colleen to talk a little bit about the partnership between LCRF and ALK Positive.
Thank you, Emily. And thank you, ALK Positive, for having us join you this evening. I'm so excited about the project. I'll just take a moment to tell a little bit about myself. I was diagnosed 8 years ago with positive lung cancer. I'm still on my first treatment. I entered a clinical trial at MGH, and I'm living with lung cancer and enjoying my life. I'm also a member of LCRF's board of directors and their scientific advisory board. I want to talk a little bit about the partnership that we have with ALK Positive, and I feel like I have a foot in both camps because I'm with LCRF and I'm an ALK Positive patient, and I've been a member of ALK Positive for 8 years now. You know, the partnership has really resulted in the recent selection and support of 2 very important projects. And we're so excited to partner with ALK Positive this year and with Dr. Schneider and build upon our mutual goals for the future. And we look forward to celebrating the partnership that we have with ALK Positive. I also wanted to take a minute to say that two thank yous and shout outs to people. Many thanks to the ALK Positive Volunteer Research Review Panel members who gave their time and their expertise to review and select these outstanding proposals. A very special thank you to Emily, who had a very integral part in developing the relationship with LCRF. And to all of you for making this research possible. The funding and fundraising that this patient-centered group has done to bring these projects to fruition is nothing short of amazing. And I guess I'd like to close and just say on a personal note what it means to me. You know, the investment in projects such as these truly hold the promise of impact in the near term as well as the long term for all of us. And if we can accelerate the pace of research, then we'll increase survivorship for those of us living with lung cancer, for those of us we love who are living with it, giving us the gift of time. So again, thank you so much for having us here this evening, and really looking forward to the presentation. Thanks. Thank you, Emily.
Thank you, Colleen. And thanks to LCRF for really such a great partnership that we hope will continue for many years. And now it's my pleasure to introduce our awardee and presenter tonight, Dr. Jamie Schneider. And Dr. Schneider is a medical oncologist at Massachusetts General Hospital Cancer Center and an instructor in medicine and a lung cancer researcher at Harvard Medical School. She received her bachelor's degree in biology from Northwestern University and her MD and Ph.D. in cell biology from Albert Einstein College of Medicine. She trained in internal medicine at Mass General Hospital as part of their physician scientist program, and then did further oncology training in the Dana-Farber Mass General Harvard Medical School fellowship program. Dr. Schneider has received grants and awards for her work from organizations including the EGFR Resistors, a Breath of Hope Lung Foundation, and the IASLC. So Dr. Schneider, thank you so much for being here with us on a Sunday evening, and we're looking forward to hearing more about your research.
Perfect. So let me pull up my presentation. Can everybody see my slides and hear me okay? I'll take that as a yes.
Yes.
Emily, give me a yes. Okay, perfect. Wow. All right. Well, what a pleasure it is to be here. And Emily, thank you for the introduction. You clearly did your homework. Everything was spot on. So thank you for that wonderful introduction.
You're welcome.
So yes, good evening, everybody. It is a true pleasure to be here. During the introduction, I was sort of looking through the names on the participant list, and I recognized several names. So hello to those of you guys who I do know directly personally, and hello to all of those of you who I don't know personally. But thank you really so much to the organizers of this incredible group for inviting me, giving me the opportunity to update you on some of our very exciting work. So as you heard from Emily, my name is Jamie Schneider. I'm a thoracic oncologist at MGH Cancer Center, part of the incredible thoracic oncology group there. And I have to say I've had the true privilege of training under some of the greats in ALK positive lung cancer. So some of you may know already, but my primary clinical training was done under the one and only Alice Shaw. I also trained with Justin Gaynor and Jessica Lynn. But I'd say that perhaps what makes me a little bit different from some of my predecessors is that I'm also a basic researcher and sort of as basic as it gets scientifically, with lab space at Harvard Medical School. And I'm really excited to tell you guys about my work, and sort of the title of this talk is I'm going to be focusing on identifying metabolic liabilities in ALK positive lung cancer. Here are my disclosures. So let's get going. So tonight I thought we could review all of the cellular metabolic pathways in meticulous detail. I can't see everyone's face, but hopefully some of you are laughing or smiling a little bit. So I know metabolism is a new field for many of you. So I am really going to start from the beginning. I'm going to go through some very basic background about why we study metabolism, and heck, what is metabolism?
Dr. Dan?
And then I'm
I'm just
and
trying
then I'm
to.
going to tell.
I'm leaving.
Yep, and then I'm going to tell you about sort of how our work in metabolism has translated to some exciting findings in the ALK space. So let's get started with some of the basics. What is a metabolite? So technically it's defined as any organic molecule that's detectable within the human body. That's a small molecular weight, less than 1,500 Daltons. And generally we think of these in buckets, right? So biologists think of sugars, we think of amino acids, building blocks for proteins, we think of lipids or fats, we think of nucleotides. All of these things are metabolites. And so some common metabolite names that you might have heard before, things like glucose, pyruvate, lactate, ATP, right, the fuel for the cell, fatty acids, DNA building blocks, these are all metabolites. And so I'd say that we, as a cancer community, we're used to really thinking about the genome, right? We don't really think so much about the metabolome. We think about the genome. Nowadays, we routinely and systematically profile for genomic alteration. So that's sort of highlighted in the pink part of this pyramid of life chart. So if you're a little bit more sophisticated, then you'll also think about the proteome, right? So we think about, cancer biologists think about the proteins that are translated from these genes and how these proteins carry out functions inside cells. But sort of the final layer to this pyramid of life, if you will, that sometimes is overlooked is the metabolome. And if you think about it, metabolite handling is sort of why genes and proteins exist in the first place. So genes and proteins are essentially needed to biochemically convert molecules in order to provide enough fuel so that cells can grow and divide, and so that organisms can reproduce. And so we talked a little bit about metabolites, but how does that sort of apply and translate to cell biology? Why does a biologist or a doctor, why do we care about metabolites or metabolism? Well, the study of cell metabolism is looking at the series of very complex, highly regulated biochemical reactions that take place inside cells. And so I sort of like the quote, cell metabolism is nothing if not complicated. So, cell biologists and cancer biologists and people who study metabolism were often thinking about how are these metabolic reactions turned on and off? How are they sped up and slowed down? How does a cell coordinate and prioritize and triage reactions that need to happen to meet the needs of growth and proliferation? How are all these metabolic enzymes that regulate all of these reactions? How are they regulated themselves? And how do metabolites control cell fate, differentiation, migration, expression of genes, epigenetics? And then why and how are metabolites secreted? And how do they impact the surrounding cells and tissues that they're in? So lots of really interesting questions that people ask in the realm of cell biology and cell metabolism. But why do we care about cancer metabolism? Isn't it just cell metabolism? So no, actually, the study of cancer metabolism is very different. Here, we are really trying to understand how cancer cells exploit metabolic pathways differently than normal cells to sustain growth. And I took out some slides, but I think it's a really interesting history, the history of cancer metabolism. It dates back over 100 years. So people like to cite Otto Warburg. He was literally around in the mid, I think, 19-teens. And he was the first really to identify that cancer cells use fuel differently, that there's something very different about how cancer cells metabolize different nutrients and use different pathways to produce fuel so that they grow and divide. So now fast forward literally a century, we have a lot more knowledge than sort of that basic start. But we know that metabolic networks support cancer cell survival, proliferation, and malignant progression in ways that differ from regular cell metabolism. We know that cancer cells take up a huge amount of nutrients, so things like glucose and glutamine, and they catabolize them differently than normal cells do. And that cancer cells use, that they depend on these intermediates of glycolysis, PCA cycle, and they use these preferentially for building blocks to build amino acids, nucleotides, lipids, so that cells can grow and divide. And so how do we actually measure metabolites in the lab? What is actually our workflow here? So I won't get too technical, but I think it's important to sort of have a conceptual background of what I'm talking about when I say, oh, we do metabolomics, we do a lot of metabolomics in my lab. But what does that actually mean? So metabolomics, sort of, I love this review from Josh Rabinowitz, published a couple years ago in Cell, sort of the metabolomics workflow, meaning how we study metabolites in cancer cells, has sort of four main parts. So we think about number one, metabolite extraction. So we can extract metabolites from various different sort of tissue types. We can extract metabolites from, here it said biofluids, but basically like serum, right? Blood samples. We can extract metabolites from cells and media. So most relevant here is patient-derived cell lines. And we can extract metabolites from tissues. So again, most relevant here for the talk today is going to be normal lung tissue and also tumor tissue. And then basically, once you have this extract of metabolites, you separate the metabolites based on principles of chromatography. I won't get into the technical details here, but once you have resolved metabolites, you run them through a mass spec machine or mass spectrometer. In Marcia's lab over at Harvard Medical School, we have 3 mass specs, all very different with different sensitivities and different resolutions. These are like multi-million-dollar machines that we run. And then on the back end, once the data is spit out, there's sort of actually a lot of work to be done on the back end in terms of data analysis. So that's sort of our workflow when we're talking about measuring metabolites in tissues. And so applications of metabolomics, I just wanted to add this in because they go beyond but they go beyond medicine, actually, right? They're sort of part of basic biochemistry. They're really important in agriculture. People who study microbiomes think about metabolites all the time. So there's a lot of really different applications in which we can apply metabolomic studies to, and cancer is certainly one important one. And so going back to why I'm interested in metabolism. So there's this, like I mentioned, this long-standing desire to take therapeutic advantage of dysregulated metabolic states in tumor cells. Tumors behave differently metabolically. How can we exploit that? That's essentially the crux of my work. And so this is already happening in real time. People have been working on this before me. And so there are multiple diagnostic tools and therapeutic approaches, some approved already. Actually, FDG PET is a metabolic assay, actually, if you think about it. But several of these are in clinical trials too. And really the overall goal of these diagnostic tools and therapeutic approaches is to exploit metabolic reprogramming in malignancy, indicating to all of us that modulating or manipulating cell metabolism is really clinically actionable. And so I took this table from a review, a really nice review in Nature that summarizes approved and then trial compounds that work through modulation of metabolic processes. And so given sort of all of this increasing data that we've had over the last 10 years or so in lung cancer, that fuel utilization preferences are different in lung cancer, there's obviously tremendous interest in exploiting these metabolic vulnerabilities for therapeutic purposes in lung cancer. So for instance, the figure on the right comes from another review where they discuss rational metabolic therapies that target unique metabolic dependencies. There are things like glutaminase inhibitors, mitochondrial complex blockers, blockers of transporters. So everyone's sort of looking for these Achilles heel of different cancers and trying to understand whether we block that particular metabolic pathway or flux down a pathway, whether we can really make a difference therapeutically. And so sort of narrowing down my talk, there's a lot of interest in thinking about cancer metabolism across different cancer subtypes. And I'd have to say there's a lot of enthusiasm in lung cancer, right? Specifically in lung cancer. And so in recent years, probably over the last decade or so, since I've been in training and then now post-training, there's been a lot of studies that have come out in very high profile journals, Nature, Science, Cell, and these have really sought to define metabolic dependencies in lung cancer, specifically in non-small cell lung cancer. Actually, I do have to say in small cell, we're seeing more and more metabolic studies as well, but sort of the focus of the field of late has been non-small cell lung cancer. And so these are studies that have been done in cell culture models, in mouse models, mainly of KRAS-driven tumors, and really some elegant work out of Texas from the Bernardinez group, who's been looking at doing in vivo isotope tracing in patients with lung cancer. So we really do have a lot more information about how lung cancers reprogram for metabolism. But you guys and I know, right? We know is better than anybody that not all lung cancers are created equally. A lot of these studies were sort of lumping lung cancers together, but we know that's not really right? So there's this tremendous molecular heterogeneity, of course, especially within the subtype, histologic subtype lung adenocarcinoma. And then of course, this matters, right? So I make this pitch, I'm a clinician, I see patients, I make this pitch to my scientific counterparts. These molecular subtypes matter because we treat patients differently depending on what oncogenic driver one harbors. And so when I started thinking about what questions I wanted to answer in the lab, I came up with sort of two overarching questions. One, do specific oncogenotypes, so whether it's EGFR mutant lung cancer, ALK-driven lung cancer, ROS1-driven lung cancer, do these different oncogenotypes confer metabolic dependencies? Nobody has looked at this. And then two, does metabolic reprogramming or rewiring play any sort of role in mediating therapeutic resistance, relapse on TKIs? And so this was a big question in the field, and this is again sort of the crux of my work. Some people have been trying to answer this really in the context of KRAS-driven lung cancers, whether specific oncogenotypes harbor metabolic profiles that confer liabilities. So for example, this was a really nice paper in Nature that came out a couple years ago where they looked at, again, KRAS backbone, KRAS mutant and LKB1 mutant lung cancer cells. And they found that there was a metabolic liability there marked by altered nitrogen handling. And I won't go into the details. It was mediated by CPS1 expression. But the bottom line is that this was sort of the first paper that I found in the literature that looked at this, that they said, okay, these KL cancers, people who have KRAS LKB1 mutant cancers, they do have unique metabolic liabilities that are potentially targetable. Another really nice example that I love that's also sort of recent is that loss of MTAP, some of you might have heard of this MTAP deletion confers a selective dependence on this protein PRMT5 and its binding partner. So that was a science paper that came out a couple years ago. And fast forward 5 years, there's a clinical trial now open. We actually have it open at MGH, and it's sort of nationwide. And they have PRMT5 inhibitors based off of these metabolic studies that were published about 5 years ago. So again, I think these are I just wanted to highlight, of course, outside the ALK space, but two really nice examples of how looking for metabolic liabilities or vulnerabilities in specific oncogenotypes can give us some more information that we can use to better inform us about potential treatment options for patients. And so I'd argue really that largely outside of KRAS-driven non-small cell lung cancer, metabolic vulnerabilities have not been explored in the context of oncogenic driver alterations, and we certainly do not know the role metabolic reprogramming plays in driving resistance to therapies. So let's dive into the part of the talk where I want to, the rest of it really I'm going to be focusing on ALK. And so just as a brief reminder, I think a lot of you guys know basic biology of ALK, but ALK is really only expressed during development. Native ALK is a single pass transmembrane protein. It's regulated tightly by ligand binding under normal circumstances. But in lung cancer, we know that ALK undergoes this translocation event in which the entirety of the C-terminal tyrosine kinase domain, the domain that's constantly signaling, that whole domain gets fused with a binding partner, which changes its subcellular localization. Now it's located inside the cell in the cytosol, where it's constitutively active, promotes survival, proliferation, tumorigenesis. And so we know, I mean, you guys took the poll, that there's a lot of you guys on alectinib and on TKIs in general, and these have been largely very successful. I mean, thank goodness for people like Alice Shaw who have developed all of these over the last 10 years. But we know that resistance is a problem, right? I mean, this is the elephant in the room. We know this is an issue. We know we need better therapies once resistance develops. And so we think about resistance in a couple different ways. We think about on-target resistance to ALK inhibitors, in which mutation occurs in the ALK gene itself, rendering TKIs ineffective. We know that in a very, very, very small subset of patients, we can see lineage transformation from adenocarcinoma to something else. But in about 50% plus or minus of patients, we see sort of what we say is off-target resistance, right? In which other signaling pathways are altered or upregulated to bypass ALK activity. And so, of course, now in the clinic, we have clinical trials looking at combination therapies, right, of ALK tyrosine kinase inhibitors with inhibitors of these other bypass pathways. But what I'd argue is that often these bypass pathways converge downstream on regulating several aspects of cell metabolism. So from my perspective, I thought it was very compelling to investigate whether metabolic reprogramming plays a role in mediating or driving off-target resistance. Again, perhaps allowing us to identify novel therapeutic targets. So for the LCRF grant that you guys heard about during the introduction, so I sort of had two main aims of this project, just in full transparency. I'm sure you guys all want to know. Aim number one was looking at ALK-driven, ALK-specific metabolic dependencies. And aim two was focusing on metabolic reprogramming in ALK TKI resistance. So I know I have sort of limited time, so most of the talk today I'm going to tell you about part A, and then hopefully I'm invited back, and then at some point in the future I can tell you about part B. So I'm going to get a little bit more into the science here. I hope I explain things adequately, and of course at the end I'm happy to take questions, as many questions as you guys have. I'm happy to answer them. So one of the first things we did was introduce either wild type ALK or oncogenic fusion ALK into lung cells, not tumor cells, but just lung cells. And we performed a very basic metabolomic analysis. When I say LCMS, that means liquid chromatography mass spectrometry, LCMS. It's easier for me to say LCMS. So that means basically I'm doing metabolomics on these cells. And we observed something very interesting, that when you introduce oncogenic ALK into cells that are non-cancerous, we saw this really selective increase of intracellular guanine nucleotides. So what does that mean? So guanine nucleotides are building blocks for DNA. So things like GDP and GTP are critical for cells' ability to replicate and divide. And so to summarize, multiple metabolomics experiments across multiple patient-derived cell lines, and again, a little asterisk here, there's heterogeneity, right? From patient to patient to patient, not everyone behaves the same. So in scientific studies like this, sort of what I build into my grant writing is it's very important for us to have sort of a breadth of different cell lines, so we can sort of capture that heterogeneity, so we know what we're studying is real and is relevant across multiple people. So again, to summarize, sort of multiple metabolomic experiments across multiple patient-derived lines, when we block ALK activity with lorlatinib, we saw something really interesting, that there seemed to be this block in purine nucleotide biosynthesis. The cell's ability to synthesize these nucleotides became impaired, and impaired specifically actually at a certain point, at a certain metabolic reaction within this biosynthetic pathway. So we were seeing decrease in GDP and GTP and accumulation of metabolites upstream, highlighted by the green boxes. So again, to make sure that this wasn't something just specific to cells, like does this happen in vivo, I wanted to make sure that we were actually measuring something real here. So we tested this hypothesis, whether this ALK-mediated change in guanine nucleotides occurred in, for example, mice, and then more importantly, in patients. Is this real? Does this really happen in vivo? I made a mouse model, an orthotopic mouse model with ALK-driven lung cancer. So the little mice get ALK-driven lung cancers. And what we can do is really cool spatial metabolomics, where we can look at spatial distributions of metabolites. And you can sort of see in this middle panel that the yellow indicates that there's increased GDP in ALK positive tumor cells in mice. And then we can actually see the same thing in people. There's not that many snap frozen cryopreserved ALK pre-treatment ALK positive tumors out there, but the few that we can get our hands on it in MGH cryobank, we can see that there's a huge amount of GDP and GTP in ALK positive tumors. So anyway, I hope that wasn't too technical, but essentially the bottom line is that we started to develop this hypothesis that ALK somehow modulates this particular metabolic reaction of converting GMP to GDP. And we thought this was really important and worth investigating further. So the next step here scientifically is we collaborated with one of sort of the world's experts in proteomics, Steve Gigi. He's at Harvard Medical School. He's a wonderful guy, runs a huge lab. And so we worked with him in his lab to do a phosphoproteomic screen in ALK positive lung cancer. And so the goal here was to identify new substrates that we didn't know before. So new metabolic substrates of ALK that regulate metabolism, that can explain all these metabolic perturbations that I was seeing. And so there were two critical things I wanted to do. One was to identify proteins that contain phosphotyrosine sites that are directly modulated by ALK. And this is, again, a little technical, but ALK is a tyrosine kinase. So we were specifically looking for tyrosine phosphorylation of these new substrates. And I also wanted to run a panel of sensitive and resistant lines, ALK TKI sensitive and resistant lines, with the goal of identifying metabolic enzymes that are differentially regulated when a tumor develops acquired resistance to ALK inhibition. So sort of long story short, we took a panel of patient-derived cell lines from ALK-positive cancer patients. We put them in a cell culture dish. We treated them with or without drug, and then performed this sort of rather complex proteomic protocol. We did whole cell proteomics, phosphoproteomics, and enrichment specifically of phosphotyrosine sites, so we can enrich our sample for phosphotyrosine-containing proteins. So that's what's shown here in yellow. And we had a really nice data set. We had several positive controls, for example, telling us basically that the data set was excellent. Sort of all the things we knew would occurred. And then we had a lot of new things to study too. That's sort of the bottom line here. And this whole project, again, is focused on metabolism. So I zoomed in and was really excited to see what metabolic genes or metabolic proteins came down in this data set. And so long story short, again, we had about 130 metabolic enzymes that we think are novel targets of ALK across 216 phosphotyrosine containing sites. And so to cut to the chase, there were a lot of interesting hits. I can tell you another time about the 129 other ones, but today, you know, when I first looked at the data set, the number one thing I wanted to do was look at purine nucleotides, because and metabolic enzymes that control purine metabolism, because that's sort of where my biggest phenotype was from all my metabolomic studies. And so a very interesting enzyme came out of the screen called GUK1. Very little studied enzyme. We really don't know too much about GUK1, so which has sort of made this project fun. And so what ultimately I found out is that GUK1 is critical for the conversion of GMP to GDP for nucleotide biosynthesis. And so, and again, the point here is that we found GUK1 to be a novel substrate, that ALK seems to be controlling GUK1 activity through phosphorylation. And so GUK1 is interesting for a lot of reasons. It sits sort of downstream both of de novo purine nucleotide biosynthesis and salvage. That's a lot of jargon, but basically that means that it's a very important enzyme for nucleotide biosynthesis. And if you look sort of in larger databases, about 6%, not just looking at ALK, but just across lung adenocarcinomas, about 6% of lung adenos harbor an amplification of the GUK1 gene, going in line with its role as sort of tumor promoting. So again, focusing on GUK1, this was one of our top hits from the phosphoproteomic screen. We indeed did several follow-up studies that I won't get into too much detail on, but basically that confirmed that ALK does indeed bind to and phosphorylate GUK1 at the site 74, which is tyrosine 74, which is what we identified in our phosphoproteomics. And then that actually is an activating post-translational modification. So the reason ALK does this, the reason cells want ALK to do this, is because it increases the ability of cells to produce GDP and GTP, which can then go on to help cells sustain their proliferative capacity. So this is just sort of data that shows ALK directly binds to and can phosphorylate GUK1. We know that this is an activating post-translational modification, like I just said, that when ALK phosphorylates GUK1, it augments GDP biosynthesis. And I'm going to skip over that because that's a little bit too technical. But so we were really interested. Let me see if my movie is going to work here. Here we go. People see it moving, I hope. Okay. So we were really interested to think about further, again, sort of fast forwarding, a little bit preliminary at this time, but thinking about how can we drug GUK1, right? That's going to be the ultimate goal. Once we prove that this is important and relevant in ALK-driven lung cancers and perhaps other lung cancers, how do we make a drug, right? So we collaborated with a structural biology team to do molecular dynamic modeling. And so we were fortunate that a crystal structure, actually of GUK1, already existed. And so what we gleaned from the crystal structure is that when the substrate GMP binds to GUK1, you see sort of closing of these two domains, the lid domain and the GMP binding domain in green and then in orange, respectively. And actually, a paper that had just been published in 2019 showed that certain GUK1 mutations that hinder the complete collapse of this GMP binding domain actually increase the catalytic efficiency of the enzyme. And this gray right here, I know it's moving, but this gray residue right here, that's the tyrosine 74 that we think that we have shown that ALK phosphorylates. So we sort of put together this hypothesis that ALK-mediated phosphorylation of GUK1 perhaps alters the dynamics of active site closure over GMP to somehow enhance enzymatic processivity. And so that's, this is again, a little bit technical from the structural biology side, but basically suffice it to say that ALK-mediated phosphorylation of this GUK1 changes its whole conformation and its ability to interact with active site arginines, and essentially helps protect GUK1 from like an open, floppy, non-catalytic conformation. So it sort of helps it stay closed, we think, to enhance enzymatic processivity. So again, I won't talk about this too much now, maybe the next time I meet all of you guys, but we are, long story short, working on developing inhibitors of GUK1. We have started a collaboration with the Harvard Therapeutics Core. This is super exciting. We have already completed a virtual screen, a docking screen. And so we're looking at ways to essentially block this ALK-mediated phosphorylation and ALK-mediated augmentation of GDP-GTP biosynthesis. So this is really exciting, and more to come on this another time. And then just a couple more biology slides, and then I'm going to wrap up in a few minutes and happy to take questions. But one of the things we did about a year ago that I thought was really important was to develop a badly needed tool. I really needed this phospho-specific GUK1 antibody to really look at where phosphorylation of GUK1 is happening. So we developed a phospho-specific GUK1 in conjunction with a company. We tested it. We know it works. We know it's specific. We know that if we knock out GUK1 and add back either regular GUK1 or a phosphomutant where that GUK1 can no longer be phosphorylated or regulated by ALK, tumors are smaller. So that phosphorylation site of GUK1 is important. Is it everything for enzymatic activity? No, but may it provide a therapeutic window? Yeah, maybe. And so again, the YF, this phosphomutant, when we put them in mice, when we put these tumor cells in mice, they grow, they're smaller. They were smaller at 30 days. And we know that this is all sort of ALK inhibition and GUK1 phosphomutant are epistatic, so they're sort of in the same pathway in terms of their ability to produce guanine nucleotides. Does this happen in patients, right? So as a clinician, again, this is really important for me when I discover something scientifically in cells. It's critical, right, to go back and say, is this happening in patients? How real is it? How extensive is it? Is it happening in one patient? Is it happening in every patient? And so we tested a panel of ALK positive tumors for whether they contain or express phosphorylated GUK1. Again, in these tumor specimens from TKI naive or pre-treatment patients, and we do see a significant amount of phospho-GUK1 being expressed. What's also very cool, and this is relatively recent data, is that other oncogenic kinases aside from ALK, so things like ROS1 and RET, maybe you guys have heard of, also seem to regulate phosphorylation of GUK1. So this is an exciting finding because it also sort of goes beyond ALK as well and may have implications for other subtypes of lung cancer. And then I added this in again last minute because I just wanted to make the point that GTP is important for cells for a lot of reasons. Primarily, people think it's for DNA synthesis. Also, what's kind of cool is that we've been able to show that the GTP that is produced through GUK1 is also important for activating MAP kinase activity. A lot of you guys are probably familiar with MAP kinase or RAS signaling, or right, you've heard of KRAS. So KRAS needs to bind to GTP to become active. And so it turns out that GUK1 is sort of upstream regulating KRAS through availability of GTP. And then in my last minute or two, I just wanted to mention some really cool technology that we're working on, A, because it's relevant for the ALK project, and B, because I'm trying my best to sort of expand this pipeline of spatial metabolomics beyond what I've sort of already been doing and to other sort of types of lung cancers. So one really important question is like, how do we think about tumor heterogeneity? Tumors have so many cells, and it's not just tumor cells. There's fibroblasts, there's immune cells, there's stromal cells, there's normal lung cells in there. So how do we sort of resolve all of that information? So we know there's tremendous metabolic heterogeneity in the tumor microenvironment. So sort of to get around that, or to address that caveat, what we've started to do is something we're calling comparative spatial metabolomics. And so I've done this with samples from ALK positive lung tumors. And so we can ask really cool questions. So can we define tumor metabolic state at a single cell level? Before, all the metabolomics experiment I was talking about was sort of bulk. This is actually looking at spatially, where are the metabolites in a tumor? Does metabolic state change depending on where the cell is localized within a tumor? Are guanine nucleotides that I've been talking about during this talk, are they specifically enriched in ALK positive tumor cells, phospho-GUK1 positive tumor cells? So this has been a really cool addition to the story, and we've been able to do spatial metabolomics in patient tumors, and we can see that there's really high expression seen by the yellow of GDP and GTP, specifically in tumors from ALK positive patients. If we overlay that information with where ALK is located in the tumor, and if we overlay with that information with where phospho-GUK1 is located in the tumor, we see that top correlations come out of GDP, GTP, guanine nucleotides. So again, this really sort of, it was like a slam dunk for us to show that in ALK positive tumor cells, phospho-GUK1 is critical for co-localizing essentially with GMP, GDP, GTP metabolites. And so, like I mentioned, I am putting together a couple additional proposals to sort of expand this beyond, because this pipeline is really interesting. So thinking about comparing TKI sensitive or pre-treatment samples with TKI resistant samples, and this would be a really neat way to sort of understand how the tumor metabolite environment changes in the context of resistance. So this is something I'm really excited about. And in my last slide, so like I mentioned, I don't have too much time to talk about the resistant arm of the project, but be sure that I am working on it. So, big question is, in resistant tumors, is there rewiring of metabolic pathways independent of ALK activity? And then specifically, based off of the GUK1 I showed you, the GUK1 work, is GUK1 differentially regulated in ALK TKI resistant lung tumors to support growth, again, independent of ALK activity? So that's something I'm actively looking at now. So I'm right about time. I wanted to leave about 15 minutes for questions. And And before I take questions, just a big shout out to a lot of people involved in this work. So over at Harvard Medical School, Marcia Higas is my main overarching PI. She's wonderfully supportive of this project. She's not a lung cancer person, but certainly very, very, very supportive of lung cancer projects. Ashita Diman is my technician who's done amazing work in this project. Shashi Joshi is another one of my technicians who's done a lot of metabolomics work. Kieran Kermi is a postdoc who did some of the biochemical studies on GUK1. A huge thank you to people that you guys know, the MGH Thoracic Oncology Group, Alice Shaw, Justin Gaynor, Jessica Lynn. Dr. Lynn in particular has been very supportive of this project and has really helped to get access to a lot of samples that we used for testing. And then of course, my collaborators and core institutes listed here. Funding, Emily mentioned at the beginning, I'm funded by sort of various institutes here listed. A big shout out, a big thank you to LCRF and ALK Positive for supporting this work. And most importantly, thank you to everybody on this call for being an advocate for yourself, for your family members. You guys are all amazing. I'm inspired by all of you, and I'm happy to take any questions. I know I spoke really fast.
Marc Muskavitch was going to, I think,
Okay.
help us pare this down a little bit for us.
Oh, right.
Good luck,
So,
Marc.
Thank you, Dr. Schneider, for that talk. It was rich, rich with contents, fairly complicated, but I think that you tried to drive through the key points and to bring home the key points for the talk. So let me just summarize a few things briefly. One thing is that this is extremely exciting work, and it's exciting because Dr. Schneider is looking where a lot of other scientists don't look, all right? So a lot of us look at genes, a lot of us look at proteins, but it's only recently, although Warburg a century ago pointed to the metabolism of cancer cells, it's only very few scientists who are brave enough to actually study metabolism, right? And Dr. Schneider is one of them. So what are metabolites really? Well, cells are made up, they're a complex mixture of tens of thousands of ingredients, right? There's DNA, there's RNA, there's proteins, and then there are these small molecules, this cutoff of 1,500 Daltons. A Dalton is the weight of a hydrogen atom, and 1,500 Daltons or less means these are the smallest molecules in cells. And those molecules are synthesized, they're put together and taken apart by the metabolic pathways that Dr. Schneider described. One of the things that happens to those molecules when they're made is that some of them acquire these little phosphate groups. A phosphate group is 4 oxygen atoms and 1 phosphate atom, and a phosphorus atom that's stuck onto the protein by an enzyme or protein called a kinase. And that phosphate acts as a signal to the cell that can cause that protein to be able to send information to other proteins, or tells the protein to move different places, or can tell the protein to be more stable or to be destroyed. And so this addition of phosphate groups is critical for the lives of proteins in our cells and other molecules. And what Dr. Schneider has pointed to is the idea that it may be that by altering the way in which phosphate is delivered to proteins that create guanine nucleotides. And guanine is a major building block of DNA and RNA, and it's also a major small molecule, something called guanosine triphosphate and guanosine diphosphate are major molecules that help cells send signals along other signaling pathways in addition to the kinase signaling pathways that ALK affects. And so to close this up and open up the chat questions and open the floor to questions, Dr. Schneider is doing exciting work because she's looking in places where a lot of scientists aren't, which means there's the potential that she's going to make discoveries that other scientists would not. And that by looking in a different way at our cancer cells, she may obtain insights and may have already obtained insights that she can then leverage to create new therapies that are going to basically kill cancer cells in new ways, and ways that might not be amenable to the same types of resistance, that we now experience from ALK tyrosine kinase inhibitors. So that's what I wanted to try to summarize.
Do you concur with that, Dr. Schneider?
I was going to private message you and say I'm going to bring you along to my future talk so you can summarize for me. That was perfect, spot on.
Emily and Mark, do you guys want to scroll through the questions, and maybe even you guys have some questions? But
Sure. Well, an interesting question that came from the earlier part of your talk is about why KRAS was the oncogene of choice for the earlier metabolomics and metabolism studies in lung cancer.
Yeah, so I was, using that as to set a framework for my work on ALK, right? So sort of to make the point that that people have done this or started to do this metabolic loop profile in the context of KRAS-driven lung cancers, and that we should be doing that in ALK-driven lung cancers because that's an obvious, to me, an obvious question. And so why did people before me do this in KRAS-driven lung cancers? I think numbers and models. KRAS-driven lung cancers are a big chunk of that pie chart. There are lots of cell lines available, commercially available, right? You don't really need patient-derived cell lines. There aren't that many commercially available ALK positive cancer cell lines. So, we're fortunate at MGH to have had 20 years worth of banking samples for researchers to work with. That's how we make progress. So I think a lot of people have done this in KRAS because of reagent availability. And then people importantly have done this in mouse models, again, because of mouse model availability. The ALK mouse model was published by Andrea Ventura, now like 5-6 years ago. But KRAS models have been around for a lot longer than that. So I think it's important for in the ALK field to take hints about the successes and challenges from other fields in lung cancer. And so that's sort of one of the things I've been doing.
That makes sense. Thank you. Let me see here.
I just have a quick question. The Enigma study and the spacewalk study, have those come into play at all with any of your material use?
Not really. And that's not to say that those studies aren't useful because they are. But one, problem is probably the wrong word to use, but one challenge that I encounter is to do metabolic analyses, do these sort of complicated metabolic studies that I've told you about. I need frozen tissue. So this, that has to be samples that are taken out in the OR, or in the when you have a pleural effusion and thoracentesis, they have to be immediately snap frozen on dry ice. And so right now that's not typical. That's not like a typical SOP, like standard operating procedure for how people bank samples. So at MGH, I am behind a huge effort to increasingly collect snap frozen tissue so that I can use them for metabolic studies. But that is a big challenge of metabolic work, is I need snap frozen cryopreserved tissue, and these are tissues that have to be at -80 or in liquid nitrogen for storage.
There's a question here, and I have no idea what this means, but I'll read it. Would inhibiting all GUK1 be toxic? Could you inhibit only PKUK1 or inhibit ALK's ability to phosphorylate GUK1? Does that make any sense?
Right. So I see that question from Matt. Matt must be a scientist, wherever you
It's a
are.
doctor, I believe, but okay.
Yeah, right. So perfect. Because that's the money question. That's the $1 million question. So right. So what Matt is asking is that GUK1 is a common essential enzyme. So 100% inhibition of its activity would be problematic and would not fly as a therapeutic. Thus, we are interested in specifically inhibiting the phosphorylation of GUK1 as is mediated by ALK, and now I've shown these other oncogenes at that site 74. So that sort of is our primary goal in collaborating with the HMS therapeutic, the Harvard Medical School therapeutics team, is we're trying to sort of be smart in our drug design approach to do that. However, there is sort of our second tier approach is still to find and characterize inhibitors of GUK1, because there are other drugs out there that target common essential enzymes. But if you can find a therapeutic window that's not.
Did Dr. Schneider freeze, or is it just me? All right, so I'm going to ask Mark and Emily some questions. Hopefully, Dr. Schneider will join. Transliteration, is that what it's called? Research. How long does that take to? What's the length of time for that to be beneficial to what we're dealing with as a patient?
You mean to become a therapeutic?
Yeah, how does this work?
Right. So the situation is a translational research, as it's called.
Translation.
It tries to translate the discoveries from the basic lab and put them into the clinical lab and then into clinical practice, right? So that's it translates the research from the bench to the bedside. And so that takes time. The fastest way that can help us as patients is if, for instance, Dr. Schneider discovered a pathway for which a drug already exists, right? Or is in late stage development in a biotech or pharma company that affects the pathway, the metabolic pathway that she is shown is critical for, say, TKI resistance. It could be that small molecule can then be moved rapidly into human trials. But even at that, it takes a number of years for a molecule, even once it's qualified for a clinical trial, to go through clinical trials, right?
Good way to segue us, Mark, because Dr. Schneider just joined us. But you can continue.
Sorry, my internet dropped out. Of course.
Normally it's my internet that drops off, but
Sunday night.
you're okay.
Yeah. I don't know how much did you hear before? So, but basically the point is that it's still worth exploring sort of general GUK1 inhibitors, because there might be an appropriate therapeutic window potentially.
And my question to Marc Muskavitch was transliteration research.
So how long does translational research take?
Translational research, right?
How long does translational research take?
To get this to the patient from your discovery.
Yeah, it's a good question. So, the most important thing with sort of discovery science is to prove that to the scientific community and the medical community that what we're working on is relevant and that we should care about it and that it should be funded and that we, need more samples. And so we're sort of still in that early phase, right? I mean, this is all new over the last 3 years. And like I mentioned, sort of as a clinician, I'm going to be 100% honest. I work with all sort of Ph.D. scientists in my laboratory setting. And I don't think any of them were really thinking like, oh, we should move this forward towards developing a drug. But I have been, right? Like from the moment that I realized that I think this is important, I've really been trying to push this forward. So with that being said, I found collaborators. We're starting on drug development. The process takes years, right? I mean, once we have a drug, then we probably first will get a tool compound, then we need to build on that, and then we need to do preclinical studies. So it does take a long time, but I promise that on my end, I'm doing everything I can, for this project to move things forward and to make it translational for patients and not just to leave it at a publication, right? As a doctor, that's like my ethos, is to make sure that this is going to be translated into something that's useful.
Yeah, what's amazing? Go ahead, Mark.
I was going to say another question that came up in the chat here is the question of whether this work on metabolomics can actually help with metabolic side effects from TKI treatments. And so do you think or know that ALK phosphorylation basically is dysregulating metabolism in ways that we observe in our cells?
Yes, it is a fascinating question. And not the, not my primary endpoint, if you will, right? I'm sort of looking at what makes tumor cells die. How can we thwart or stem resistance? But I have often thought about, whether or not these findings have any implications for the metabolic side effects that we do see on patients with ALK-TKI. So I think it remains to be determined, but a question that I do often think about. Good question.
Right. And then another question that was from the chat is, sometimes alternative and complementary medicine will portray cancer as a metabolic disease, whereas sort of neo-traditional science portrays it as a genetic disease. Now, in fact, in science, we know that those aren't really, that's a false dichotomy in a sense. But how do you think about how your research addresses cancer as a metabolic versus a genetics disease?
Yeah, what a great question. I am going to just spin it a little bit and tell you my first thought, which is that I agree with that statement that it's problematic, in my opinion, that we think of cancer as a genetic disease. I think it's so much more than that. And that's evidenced by when we look for genetic explanations, when people develop resistance, we often can't find them, right? In a proportion of patients, we do see mutations in the ALK gene itself, or MET amplification, or some other sort of rare genomic alteration. But that's a minority, right? And so the majority, we can't explain things genetically. Hence, why I wanted to study something sort of non-genetic. Like what are the other layers in biology that make sense that could explain the biology that we can't explain genetically? So that's sort of my take on that question. I 100% agree that sometimes we think about cancer too much entrenched in genetics, but I think there's a lot more to it.
I know we're getting past the top of the hour, and we really appreciate the time you spent. There are a couple questions that are similar in the chat that might be a good one to end on. What can patients do to help with this study? And I know a lot of us have helped already by contributing and funding the research, but is there anything else we can do in terms of contributing samples or anything like that you can talk to us about?
Yeah, I mean, you guys are all amazing. The fact that you're on here on a Sunday night, learning about science, probably a lot more science than a lot of you ever wanted to think about, especially on a Sunday night, and advocating and fundraising and helping with community awareness. I think all of those things that you guys are already doing is nothing short of incredible, and really inspiring. To help with research efforts, it's logistically challenging, right? Like we need cryopreserved specimens, we need pre-treatment, post-treatment. I'm sort of lucky and privileged to work at MGH, in which a system is set up. But I think there are people on the call not in the Boston area. I would absolutely speak to your healthcare provider about what are the opportunities locally where you're being seen to preserve tissue, whether that it's at diagnosis, for patients who are new to the group, or on biopsies that you'll have going forward. I just think it's really important to bank extra samples so that it's there. So when somebody comes along 10 years from now with another great idea, that tissue will be available to study. So.
Thank you.
Yes, thank you so much, Dr. Schneider. Even though this may have gone over most of our heads, your passion is amazing, and it makes me like, I don't care what you're telling me or what language you are speaking in, just your enthusiasm for the topic that, again, like Marc told us, many scientists and physicians won't dare to touch. Like, we're all so grateful for everything that you're doing behind the scenes. Like we talked in our pre-game, the behind closed doors, we're also grateful for the passion that you bring to this topic and your team that you have working for us. So thank you. And thank you all for contributing, Emily and Marc, and Stacy from Lung Cancer Research Foundation, and Colleen. We're so grateful to have you all here. We do have ALK 101 Part 2, Assembling and Managing Your Care Team, coming up on March 19th. So in 2 weeks, you have a little break. And then we will be reconvening. Again, you can catch part 1 on YouTube. Also, spread the word for those that missed tonight's talk. That will be on our ALK Positive YouTube channel in the next few days. So thank you all for your assist in moderating. And Dr. Schneider, thank you so much for being here and for everything you're doing for us. We're so grateful.
Thank you. Thank you so much for having me.
Yes,
Thank you. Thank you,
Thank
Doctor.
you, Dr. Schneider.
Thank you.
Thank you.
Thank you.
Thank
Thanks very
you.
much.
Thank you.
Thank you.
Thank you, Dr.
Have a good
Schneider.
evening.
Thank
Bye,
you.
everyone.
Thank you, Dr. Schneider.
Thank you.